Welcome back to Metabolism unit. We are going to talk about enzymes because after all, enzymes are the stars of biochemical pathways. As you can see here, I have a snippet of biochemical system. And notice the system is a very complex network of chemical reactions that are happening. So each dot here in the system represents a small molecule, and each line in the system represents a metabolic reaction. And each of these reactions are catalyzed by very specific enzymes. So can you imagine the complexity of what is happening here to carry out those metabolic processes within our cells? It's pretty amazing. As we learned in our biochemistry unit, most enzymes are proteins. Yes, we have a few RNA molecules that can carry out enzymatic activity, and we would call them ribozymes. However, here in this unit, we're going to be focusing on proteins. So understand that the shape of the protein is going to be super important, and the sequence of the amino acids in a polypeptide chain is going to determine what kind of shape this protein is going to have. How is it going to fold? If you recall, we have all these levels of organization within the protein that gives rise to that particular structure and therefore function. So we have a primary structure, which is the sequence of amino acids. We have peptide bonds in between the amino acids. And then we have the formation of hydrogen bonds that leads to the secondary structure. And it's either going to be alpha helix or beta pleated sheet. And then tertiary structure results because R groups of the amino acids are now interacting. So, and you can see it takes on this three-dimensional shape in the protein. And some proteins will even have a quaternary structure where we have multiple polypeptides that are folded, sort of coming together and carrying out a specific function. So because proteins will have a particular shape, when it comes to enzyme, there will be one area in the protein that's going to be really important. So we call that part active site. So active site will specifically interact with the substrate molecules. So the way they interact with each other, it would be via hydrogen or ionic bonds. And that means the active site and the substrate must be compatible to one another. So if we take a look at this example, I have an enzyme called sucrase. So enzyme sucrase will recognize its substrate, that's called sucrose. So notice sucrose will interact with the active site, and then we say enzyme substrate complex forms, and this is where catalytic reaction is going to be taking place. So there's an addition of water because after all, we have to cleave this bond that's holding these two monosaccharides together. So substrate is then converted into products. Products are released, and in this case, we have two monosaccharides, glucose and fructose. Enzymes do not get used up by the reaction, and they can be used over and over again, and they are only going to lower the required activation energy and speed up the chemical reaction. We used to describe the enzymes and substrates as if they were a lock and key, or they sort of worked like a lock and key model. So today we know that enzymes are not exactly rigid structures, that when the substrate binds to the enzyme slightly undergoes conformational change. So, and then this change is this tightening that happens around the substrate is called induced fit. So here I have an example. This will be my enzyme. And notice it's obviously a super simplified model, but you can see an enzyme active site right here. And then this would be my substrate. So you can see the substrate will fit in the active site. So there's this compatibility going on. And when that happens, the enzyme will undergo a slight conformational change. And you can see it's going to tighten around the substrate. So there it is, tightening around the substrate. So we call that process induced fit. And this process is going to enhance the ability to catalyze the reactions. So there's this tight, snugly fit around the substrate. So in this case, you can see here, I have an enzyme called hexokinase. So this is its empty site, and notice the configuration of what this enzyme looks like now. And then when the glucose and ATP, in this case, we have two substrates that are going to sit in this active site. The enzyme and the substrate will sort of snuggle together properly. And in this case, this glucose is going to be phosphorylated. So ATP will be hydrolyzed, and that additional phosphate group from ATP will be attached to glucose. So here's another picture of what induced fit or strain looks like. And then, of course, the bonds in the substrate are going to be stretched and pulled and bent, and of course this is going to push the reactants into this unstable transitional state so that way the reaction can actually proceed forward. So another way an enzyme can catalyze a reaction is basically providing this correct configuration or positioning or orientation for the substrates to come together. So when they come together, then of course the reactions are going to be happening between them. Another example of how enzymes can provide a microenvironment for the reactions to occur would be, let's say acidic R groups may provide the necessary hydrogen transfer to the substrate. So, and therefore that can actually initiate the reaction. So we say, you know, for this reaction to occur, we need a low pH, and that's why we say, oh, there are the R groups that are actually donating that hydrogen. And in some cases, we can even see enzymes forming covalent bonds with substrates, but usually those are brief, short-lived covalent bonds, because after all, we have to release the products. So some enzymes will require other molecules in order to function. So these other molecules are called cofactors, and I'm going to give you a few examples of such. So these can be metal ions, they can be organic molecules, and we call them coenzymes, and they can also be prosthetic groups, and these are typically permanently bound to the enzymes. So metal ions, few examples here, iron and copper and zinc, coenzymes, biotin, coenzyme A, NAD, FAD, even ATPs. So we can refer to these also as vitamins, and when we buy multivitamin capsules, they will usually contain mixtures of all these vitamins at different percentages. So now we're going to talk about how do enzymes catalyze the reactions. So how do they affect the rate of reactions? So we know that they are going to increase the rate of reaction because they are catalysts, and they do so by lowering the required activation energy. So if we look at this graph here, on the y-axis, we have free energy. Remember we talked about the Gibbs free energy. So these are my reactants before the reaction. So they are sitting at stable state. So the reactants must be pushed to the transitional state, which will be unstable. And then the reaction proceeds, and the outcome would be our products. In this case, notice the energy levels went down. So that means we had a larger molecule that was broken down into smaller components, and therefore we would call this reaction as exergonic. So this activation energy, this kick over the energy barrier, is represented right here. So when we use the enzymes, this required activation energy will be much lower. So analogy here would be this ball on a hill. So it's sitting at a stable state. So it means we have to invest energy in order to push that ball over the hill. So we basically push them into that transitional unstable state, and therefore this reaction is going to happen as such. If you take a look at this graph, compare uncatalyzed reaction with catalyzed reaction. So catalyzed reaction, notice the free energy, the activation energy is only this little tiny hump. You see it? If we look at uncatalyzed reaction, the required activation energy is much greater. So we have way larger hump to overcome. So, and to put things in perspective, notice that here I have one example. A molecule of sucrose in a solution may hydrolyze in about 15 days. But when the sucrose is present, the same reaction happens in 1 second. Wow, so you can see how they actually speed up chemical reactions, and then again, they do so by lowering that activation energy required. So after all, biological systems, we depend on enzymes. We would not want to depend on heat to initiate those chemical reactions, because not only all these reactions would be accelerated, but remember the destructive ones would also be accelerated. Is there a time when enzymes are saturated or become saturated? So let's take a look at this graph. So notice we have a concentration of substrate that increases, and then on the y-axis we have the reaction rate. So the reaction without the enzyme, notice there's a steady increase, but it looks like the reaction rate is much, much slower. And then with the enzyme, so this is a catalyzed reaction. So it increases very rapidly in the beginning, but then it levels off. You can see it reaches that maximum rate. Why? What is happening here? So notice what we are increasing here is the substrate. So if we continue to increase the substrate and do not increase the enzyme, So what this means, all the enzymes that we have, they become saturated. So imagine if I have an enzyme here, so I'm just going to draw a little one right here, and they all are going to be interacting with a particular substrate. So if we add more substrate, you can see how the reaction rate will still continue to be the same. This is where it levels off, because when one substrate comes in, releases the product, so another substrate can come in. So there will be this leveled off particular rate of reaction, because now all of these enzymes are working literally to their full capacity. So they are all interacting with substrates at a particular rate. So adding more substrate, no, this will not increase the rate of reaction. So now the next question that we have here is, can cells regulate metabolism by controlling the amount of an enzyme? And the answer is yes. So first of all, we're talking about gene expression. So this is what we have: DNA. DNA is expressed into mRNA. mRNA is translated into protein. So this would be your enzyme. So we can either turn on the gene to produce that protein, or we can turn it off. So that's one way to regulate the amount of enzymes we have in cells. So the cells have a certain number of enzymes, and another layer of regulation would be that they can use regulatory molecules to activate certain enzymes and deactivate them. So we call this regulation allosteric regulation, when regulatory molecule binds to an enzyme at someplace else other than the active site. So let me show you an example. Allosteric inhibition would happen when an enzyme is active. However, an inhibitor molecule binds to the enzyme allosterically. So in other words, you can see here, it's not binding to the active site, but instead is binding someplace else other than the active site. So it's binding on the other side of this enzyme, and this enzyme, therefore, will undergo conformational change, and then the active site will be no longer there. So you can see the active site now will change, and the substrate will no longer be able to interact with the active site. So it means we shut this enzyme down. So this enzyme has been inhibited allosterically. Here's another way to regulate the enzyme. So this would be allosteric activation. So suppose the active site right here is not compatible with the substrate. So when an activator molecule binds allosterically to this enzyme, the enzyme undergoes conformational change, and now this change provides a compatible active site for the substrate. So this enzyme is now functioning, but this activator is the one that's binding allosterically to it and causing that change in the active site. Another type of allosteric activation would be a cooperativity. So this is where we have multiple polypeptides that come together into one big globular protein. And then if you have only one substrate that binds to one of the active sites, that causes the conformational change in the rest of the polypeptides. And notice now their active sites are going to be compatible for the substrate. So it will have, the substrate will have high affinity to the active site, and therefore they will bind, and this enzyme is fully active and functional. Cells also can use feedback mechanism to regulate the enzymatic activity. So this is where we have a buildup of end product that can act as allosteric inhibitor and inhibit the metabolic pathways. So an example here, we call that actually negative feedback mechanism. So here we have threonine as starting material, and you can see there are multiple reactions that are happening. And then the final product would be isoleucine. So this is the end product, which when the levels of isoleucine rise in the cell, isoleucine will act as an allosteric inhibitor of one of the enzymes here to shut down this pathway. So you can see now none of these reactions are going to be happening anymore, because the cell is basically saying, okay, we have enough of that end product. We have to maintain the specific homeostatic levels of that particular product, and that's why the pathway is going to be shut down. So certain chemicals can inhibit action of enzymes, and this inhibition can be reversible and irreversible. So I'm going to give you an example of reversible inhibition. Competitive inhibitors are molecules that are going to be competing for the active site. Remember, we've talked about allosteric inhibitors, but now we're talking about competitive inhibitors, because their shape will resemble the natural substrate. So it means they have very similar shapes, and they both can actually fit in this active site. So competitive inhibitor, when it occupies the active site, obviously that renders an enzyme to be useless because no reactions are going to be taking place since the substrate can no longer bind. A non-competitive inhibitor, we actually talked about that already. So this is allosteric regulation. When you have a molecule that binds allosterically to the enzyme, causes a change in the active site shape, and therefore the substrate can no longer bind to it, so it means no reaction is going to be taking place. And then we have irreversible inhibition. So irreversible inhibition happens when a molecule binds covalently to the enzyme. So an example here is I have an enzyme called acetylcholinesterase, and here is its active site. So we produce insecticides and include this chemical that is going to be compatible with the active site. Acetylcholinesterase is an enzyme that plays a role in the nervous system. So it removes neurotransmitters from the synaptic cleft. So for example, in insects, when the neurons release neurotransmitters, these neurotransmitters would initiate muscle contraction in the insects. So the neurotransmitters then have to be removed from the synaptic cleft to stop the muscle contraction. So this enzyme actually does exactly that. So its job is to remove those neurotransmitters. However, if we inhibit this with a chemical that binds to the active site, that means this enzyme is no longer working. So a poor insect, when sprayed with the insecticide, will continue to release neurotransmitters, but then cannot clear it out of the synaptic cleft. So the muscle twitching will continue until that insect dies. So now what I want you to keep in mind is that cells carry out selective inhibition. Do not think of molecules that can inhibit enzymes that will always be poisons. No. Cells can actually make specific molecules to inhibit them because we're talking about reaching those homeostatic levels of specific products. So it means cells will do whatever it takes to be able to turn on the enzyme, to turn it off, because the goal is all that homeostasis within the cells. Enzymes are going to be affected by the environment because after all, we said they are proteins. So if the environment changes, it can affect their 3D structure and we can actually denature the enzymes. So when we say an enzyme got denatured, that means it lost its secondary, tertiary, and potentially quaternary, if there was one, structures. So if conditions have been restored, sometimes we can renature them. They can fold back into that normal shape. So few environmental conditions here to explore would be the pH. So pH is going to affect ionic bonds and hydrogen bonds. And then remember that all enzymes will have their optimal pH for their activity. And usually that range is pretty narrow. Temperature changes, increases in temperature obviously supply kinetic energy. So molecules are going to be colliding faster. So therefore you're going to see the rates of reactions go up. But then when the temperature drops, kinetic energy is lowered, so it means the protein is not going to get denatured, everything is just going to slow down. But going back to the increase of the temperature, we can actually reach that maximum rate, and if we continue increasing the temperature, we can denature that protein because there will be more vibration, and that's going to lead to the breakage of hydrogen bonds and loss of that 3D shape. Alcohol can also disrupt hydrogen bonds and compromise its shape. And then metal ions as well, they can actually disrupt ionic bonds and disulfide bridges that happen between cysteines. And remember, those are your strong bonds that hold that 3D shape in a protein. And the last piece of information, I thought it was pretty cool because everybody has heard about aspirin and you have heard about penicillin, which is antibiotic. So aspirin happens to be an enzyme inhibitor. So it inhibits a specific enzyme, and this enzyme is involved in the production of prostaglandins, and these guys are involved in producing inflammation and pain in the body. And then another one is thromboxanes. So these will lead to blood clotting. So if we inhibit this enzyme, this is what aspirin does, so it means we're going to eliminate those symptoms. Another example would be penicillin. So this is also an enzyme inhibitor. So this enzyme inhibitor will inhibit transpeptidase, which is an enzyme that bacteria uses to build bacterial cell walls. So if we inhibit this enzyme, that means bacteria can no longer build their cell walls, they cannot divide, and therefore we are going to get rid of them and clear the infection out of the body. And this will complete our discussion about the enzymes. I will see you in the next biochemical pathway, which is going to be cellular respiration.